Thank you so much, Dr. Tsakalozou and Dr. Sharan. And we actually have another speaker joining us for the panel. It's Dr. Lucy Fang, who is the Associate Director in the Office of Research and Standards in the Office of Generic Drugs. So welcome, Dr. Fang. And with that, we're going to go ahead and get started with our question and answers. If you have questions, please put them in the Q&A chat pod, and we will get to as many questions as we can before lunch. So with that, our first question is going to be for Dr. Tsakalozou. Could PBPK modeling and simulation approaches be applied to support development and approval of all locally acting products? What are the added advantages of employing this in silico methodology for generic locally acting products?
Thank you very much for the question. I hope that you can hear me well. I tried to highlight in my talk and answer this question. The added advantage of using in silico methodologies is that they can provide us with an insight information on what is happening, what is the concentration of the active ingredient at the site of action. So for locally acting drug products, it is not feasible most of the time to access the site of action. It can be in any of the skin layers. So with modeling and simulation, we can actually predict what would be the drug amount in that specific skin layer, which may be the site of action or may be very close to the site of action. So that is the added advantage. Another advantage that I can think of is that we can extrapolate our predictions from healthy skin to diseased skin, assuming that a disease can be properly characterized in the model. And another important consideration, especially for generic drug products, is that the product quality attributes can be taken into account within those predictions. So if there are differences between the reference and the test product, these can be are reflected in the predicted local bioavailability, and the quantitative character of PBPK modeling allows us to understand what is the magnitude of these differences and assess that within the bioequivalence context.
Great, thank you. And we have another question for you. What is your view or FDA's view on alternatives to human skin membranes in permeation studies? For example, pig.
So this is a good question, and we know that animal studies are used preclinically towards the development of a product. We need to, from the modeling perspective, we need to be aware of the differences in the skin physiology between pig skin or any animal and human skin. So there's a question on how a model that is developed with pig skin data can actually be translated to human. So the physiology is one component that is not in agreement, and it is important, and it is in direct connection with the disposition of your active ingredient in the skin. So the way the permeation takes place for an active ingredient within the skin of a pig may be different from skin permeation in humans. So these are considerations when developing a model and when using animal studies towards product development. I would say that at early stages of development of a product, pig studies can be useful. But when it comes to actually answering a bioequivalence question, we do not accept animal studies. We are focused on human studies. They're more informative.
Thank you so much. And we have another audience question for you. Does the PBPK model account for the dynamic desquamation process of stratum corneum cells? Does the model also account for the drug and keratinocytes?
These are both very important physiological processes, and for some drugs, for some active ingredients, from some drug substances, they can be very important. So I would say the way I would approach this question and expand on it in terms of developing a model, if you think that that this process is important for your drug product and for your drug substance, then it should be included into your proposed model, because it will impact local bioavailability, it will impact in vivo performance for your product. If it is not important, then it can be included into the model. The question is, the challenge here is how these processes can be incorporated and informed into the model, informed by the model. So is there experimental data available that can support the model structure, that can support the selection of the parameters when building a model? So these are questions that need to be answered as we're developing a model that accounts for these physiological processes. And again, if these are important for a specific product or a specific drug substance, then they should be accounted for in the proposed model.
Thank you so much. And with that, we're actually going to go have Dr. Sharan, if you could take some questions. So the first question to you is, can model-informed and model-integrated approaches be applied to other categories of products other than long-acting injectables?
So thanks for the question. And the short answer is yes. And as I stated during my presentation, that using model-informed and model-integrated approaches can help in designing an efficient clinical bioequivalence studies. And if you think that essentially can be applied to other product categories as well. However, the impact of using these approaches can be more widely seen in long-acting injectable products, because conducting a clinical B studies is very challenging because of very long half-life of these products. Hope that answers your question.
Yes, great, thank you. And we have another question for you from the audience. Where can we find more details regarding the work being done by Carlson to help guide clinical development plan considerations?
Been working with the University of Uppsala on these model-informed integrated approaches for long-acting injectable products. And Dr. Matt Carlson, as well as Dr. Andy Hooker, who is the co-PI for this contract, they have presented in the past and scientific meetings like American College of Clinical Pharmacology in 2019, and in the American College of Pharmacometrics. And also, we will, once we have an outcome for this contract, we would be publishing a manuscript with details on how these studies can be conducted. Also, we are also working on, through this contract as well as another contract, to develop and provide an user-friendly, easy-to-use RSANI package that will incorporate both non-compartmental analysis-based B analysis and model-based B methods for situations where conducting these non-compartmental analysis studies is practically challenging. And this R package can also help in supporting alternative study designs, reducing sample size, or reducing study durations for products, as discussed talk today.
Thank you, Dr. Sharan. If you can, I think you sound very clear and good, but if you can even come a little closer to your mic, I think that will help our transcriptionist make sure that she captures everything. And we have another question for you. Could you please clarify whether switching study with attainment with reference product and only single dose of a test product as part of period 2, can that be?
So this is a novel study design which has been proposed by our grantee from University of Uppsala, Dr. Matt Carlson group, and we are working on that study design, and the group is still evaluating that proposal, and if successful, we would be providing more clarity and more information on that through publications as well as presentations in that regard.
Thank you so much. And now we have a question for Dr. Fang. Can you comment on whether PBPK modeling can be used for virtual bioequivalence simulations of long-acting injectable products, in addition to pharmacometric?
Can you hear me? Okay, thank you. Thank you for that question. So the answer to that question is yes. As long as the applicant can adequately validate the PBPK model, such as characterizing the different levels of variability, such as the between subject or the within subject variability. So if the applicant has a specific modeling question, we encourage the applicant to submit a pre-ANDA meeting package to FDA. Thank you for that question.
Great, thank you. And we have another question for Dr. Tsakalozou. What are the key considerations on implementing PBPK modeling and simulation towards development and approval of a locally acting product?
To answer this question, we need to keep in mind what are the challenges of developing and validating, especially a PBPK model for locally acting drug products. So, as I mentioned in one of my previous answers, the main challenge with locally acting drug products is that we cannot access the site of action. So when we are developing a PBPK model, we are taking into consideration the local physiology, and that is accounted for in the model structure. But when we are but what the model is actually providing us with is a quantitative description of the permeation of the drug substance through the skin. And the main question that we have as this model is developed and applied is how reliable, how accurate are our predictions. So the basic question is, how do we validate those predictions? How can we compare the model predictions with actual data, observed data? This data, as I mentioned in my talk, can be obtained by IVPT studies. They can be obtained by microdialysis or open flow microperfusion studies. So studies that allows us for an insight on local bioavailability. So this is a big challenge. And then the other it is a challenge because we need to be certain of the quality of the data and the integration of that data within the model in terms of development as well as validation. The other challenge is that we usually do not have systemic exposure data for, especially for topical dermatological products. So the only validation that can take place for our model when we are developing it is locally within the skin. So this is a consideration. Then if we are developing a model for a diseased skin, potential interplay between a drug product and the diseased skin is also a question. And the other final consideration I'd like to bring to the table, and it was not highlighted in my talk, is the population that we will decide to run our prediction, to run our simulation and ask a bioequivalence question. Is it going to be healthy volunteers? Is it going to be healthy or diseased skin? What is are we going to recruit only male or female subjects? So all of these, what is the variability for each one of these populations? So these are all questions that we need to consider as we move on towards developing, validating, and applying a model for its intended purpose.
Great, thank you for that answer. Our next question is for Dr. Sharan. Do you think FDA will consider these models as standards in specific bioequivalence requirements?
So as presented in my talk, for long-acting injectable products, conducting the conventional PKB studies are very challenging, especially if you look at half-lives, some products which have half-life ranging from 3 to 6 months. So for those products, FDA is encouraging applicants to use innovative approaches, which includes modeling and simulation. And that can be used as an alternative approach to justify your study, and it can be used for this.
Great, thank you so much. And we have another question for Dr. Sharan. Jumping back up. What is the status of the modeling contract with the University of Uppsala, and what are your plans to disseminate this information to the generic drug industry?
So this question I partly answered earlier, but I would repeat that this contract was awarded in last year in May 2019, and this contract is in the middle of the performance, and it would be completed next year, and we intend to disseminate the outcomes from this contract via manuscripts as well as presentations at international scientific meetings like American College of Clinical Pharmacology or American Association of Pharmaceutical Scientists, or as well as SBIA meeting like this, and through manuscripts as well. So that would give an opportunity for the wider audience to see the approaches and utilize in their product development plan.
Okay, great, thank you. And our last question is for Dr. Tsakalozou. What are the objectives of the research funded under GDUFA 2 in support of modeling and simulation methodologies for locally acting drug products?
Thank you for this question. I think that during those 2 days of a workshop, most of the speakers have presented outcomes of the research. So the aims, the objectives of the research are twofold, essentially. We are trying to acquire data, experimental data that will help us answer specific questions. These may be related to, they're mostly related to product-specific guidance development, and also they can be related to ANDAs for complex drug products. The locally acting drug products are considered complex drug products. However, in addition to bridging the knowledge gaps that we have identified as an agency in collaboration with the generic drug industry, we are also using GDUFA funded research to develop modeling and simulation tools. These modeling and simulation tools, when they are combined with the data, the knowledge that will be acquired by the rest of the GDUFA-funded studies, can actually help us have in hand tools that will support alternative B approaches. And these alternative B approaches can help reach approval for complex drug products. And through the GDUFA research, that's my last sentence. And through the GDUFA research, we are trying to disseminate these outcomes, as Satish also mentioned, to the public, so that the industry can also leverage all these modeling and simulation tools. So our goal with GDUFA is to bridge the gaps and work with the industry to develop modeling and simulation tools that will help us support alternative B approaches.
Great, thank you so much. And with that, actually, we're going to ask Dr. Sharan to share the poll results from the presentation earlier. And if you can just speak very loudly and close to your mic, that would be great for our transcriptionists.
So thanks for everyone who participated in the poll. And based on the polling results, we saw that around 70% of the people who polled have at least considered or are thinking about using these model-informed and integrated approaches in their bioequivalence study analysis plan, as well as for designing those studies. And a few, I think 30% of the audience have stated they have not considered, but I hope that this presentation would help them to educate on how they can leverage these innovative methods for their product development plan. And if you can also look at the presentation, or you may have attended Dr. Robert Leinberger's, our office director's talk yesterday, and he also indicated that how you can use these innovative modeling and simulation approaches for like the B studies, which has been interrupted due to COVID. So this is really promising, an innovative approach, and I hope, based on the polling results, at least majority of the audience are thinking about applying these innovative approaches in their B study design.
Fantastic. Great. And with that, we are going to take a break for lunch. We hope that everyone will join us at 1:05 for the next session, which is Practical Considerations in the Study Design and Data Evaluation recommended in Product-Specific Guidances. So we will see you back at 1:05.